Okay, Addy, I think we can start. People may come in.
Perfect.
I'll
Hi,
just
Stefan.
explain. Hello, Stefan. Hello, Jacques, Livio. So just quickly to explain that at the bottom right of your screen, you will see a little icon that says interpretation. If you click on that, you'll see a choice between off, English, and Spanish. If you click on English, you will hear the presentation from our interpreter, George. Otherwise, the main presentation will be in Spanish. So if you want to click on that icon and switch to English, you will then hear the English translation from Spanish. Thank you. Okay, Addy.
Bueno, bienvenidos a todos a este webinar sobre tecnología de clonación de animales. Semanas atrás, cuando lo conocí a Gabriel Vichera, yo le comentaba que esto que hacen desde Kheiron es algo así como ciencia ficción, algo que cuando éramos niños realmente lo veíamos en las películas y realmente esto está pasando. Ellos desde Kheiron lo que hacen es clonar caballos de polo de excelente calidad, de excelente estirpe, y con ello lo que logran es una mejoría increíble en estos nuevos clones. Pero bueno, quiero darles la bienvenida y agradecerle al doctor Gabriel Vichera para que nos cuente realmente qué es lo que hacen desde Kheiron con la clonación de animales.
Hello. Thanks for the. As of now, I will continue in Spanish. As I said, the main service or product in our laboratory, Kheiron Biotech, is clones. We produce clones as a service or product. Okay. Please go ahead.
No te escuché, Gabriel. Sí, por favor.
No, simultáneo, simultáneo.
Simultáneo.
Yes. Live. We produce clones as a service, as a product. But let's begin with the definition of what is a clone. A clone is an organism that is genetically equal to another, that is made out of a asexual reproduction. Later on, we're going to see how this is done, the technical side. The first clone that was produced of an adult was the well-known dolly. This was the first clone that was done as an adult. That's why this was considered a biological revolution. This broke a paradigm. Until then, scientists thought that you could not produce a clone from differentiated cells. So this was a true revolution. And many scientific movies, sci-fi movies, this opened up a new world from beyond the science fiction. The dolly breaks a paradigm. As of 1996, where Dolly is cloned, first clone as of an adult. Then many laboratories around the world started to clone other mammals. And then through the following years, in 98, first clones of mice, first clones of cows. Then pigs were cloned in 2000, 2002, cats were cloned. 2003, something that we were very interested about here at Kheiron. Horses, the clones were made, a horse and a mule. In 2005, we arrived at dogs. And then the species continued to expand over the years, the clone species. Recently, in 2018, the first clones of the first primate, non-human primates, of course. But this also brought forth a controversy. But if you develop a protocol to produce a clone of a primate, which are very close to humans, more than 95% of the genome is identical. We believe that using the same protocol, you could clone a human. But the question lies in why clone? What are the applications to cloning? What are the advantages of cloning animals? The first of them, the most intuitive, is to multiply and replicate animals with certain differential characteristics. For example, in cattle, if I have a cow that has strong attitudes for producing milk or better meat, what I can do is make copies of those founders in order to quickly disseminate those characteristics, those special characteristics in a herd. That would be one of the main advantages of cloning. Another important application or advantage of cloning until today has been the application of cloning in the conservation of species and recovering extinct animals. On the left, in 2003, there was a bucardo, which is a subspecies of goats, that was cloned in 2003, which was an animal that was extinct. This clone was born, and the animal died. So we extinguished this animal the second time. Then the central picture, 2020, is a Brzezowski horse. It's a very primitive horse. Civet in 2021. We were both cloned recently. We produced clones. Not Kheiron, this is another company, BHM. They did this to multiply the individuals. Then animals that are in extinction or in danger of being extinction. This is another application of cloning, the conservation of endangered or extinct species or recovery. A third application that has many variables is to apply modifying editing or modifying genetics in animals. Cloning gives this advantage that you can produce genetic modifications in a cultivation of cells to make sure the cells have all the modifications that you want, and as of those cells, produce embryo clones. And then those embryo clones can be born cloned. Here we have the example of these cows that were produced in Argentina. They're transgenic farm cows that express in their milk growth hormone. Human growth hormone is produced by their milk. It's a pharmaceutical that produces the pharmaceutical inside the animal, within the milk. This is an example of what we can do with cloning to modify animals. Another example, xenotransplants. This is a research line that we also have at the company. As I was saying, through cloning, we can edit genetic cells, and those cells can be checked and then adjusted and produce animals of those cells. This is a great advantage for xenotransplantation. A xenotransplant is a transplant of an organ from an animal to another animal, for example, a human. So we are talking about producing organs that are compatible with humans from animals. Now, why? For example, in the case of the pigs, the organs are physiologically similar to human organs in terms of size, a heart, a kidney. But the human body, when it receives a tissue or an organ from an animal, recognizes that as an strange body because of its antigen, and the human body rejects the transplant automatically. What is called a hyperacute rejection, and eliminates the tissue or the organ. So, through genomic editing and combining with clonation, we can make those organs produced that are not rejected. We can block the expression of certain genes which produce the rejection, and we can humanize certain genes. We can rewrite their genetic code so that those proteins are similar to human proteins, and thus we avoid this immunological response and rejection. This is another advantage of cloning. You can do the editing themselves, but then to produce the animal, it's very useful to use this cloning technique. Another advantage of cloning, combined with modification of genes, is, is to give certain special characteristics to the cattle. You can produce cattle with resistance to illnesses. You can improve the quality of the meat. We're doing this currently at Kheiron. What we are doing is modifying the expression of a certain enzyme, which makes the cows to have a higher content of protein. So they will produce more protein, consuming less. Other characteristics we can obtain is a better sustainability of the animal. In the lower picture, we see a calf that was made through genetic editing, has shorter hair. And what's the advantage of a shorter hair? That it resists higher temperatures. It doesn't suffer heat so much. So this animal may be bred in more adverse climates, for example, in warmer climates. So you can create cattle with better characteristics of meat, which resist higher breeding temperatures or places where temperature would be more difficult for the animal. This is another advantage of cloning. Another potential application of animal cloning, which would let us, as a company, is to produce copies of elite race horses or sports horses or even pets. We primarily make copies, genetic copies of horses, of elite sports horses. Our first clone was born from a racehorse or a sports horse, a polo horse, in 2013. 2013, the first clone of a polo horse was born here. Today, we have cloned and given birth to more than 200. makes us one of the most productive labs in the world in cloning. Why are so many clones made for horses, for sports horses? Why sports horses? Because I have like a photocopier, which is what a clone lab is a photocopier of animals. In the case of polo horses, produces a lot of advantages for the breeder. For example, in Argentina occurs the following. The most important tournaments of world polo are played out in Argentina. They're played in the springtime. Springtime coincides with these tournaments and the reproductive cycle or period of the mares. So what does this mean? When the mares are playing in the best tournaments, they can't be reproducing and having offspring. So traditionally you had to wait until the tournaments were over, wait for summer, which is not the best time to reproduce the mares, and maybe they would have one offspring a year. By cloning has given the breeders the opportunity of having mares competing and their copies reproducing and producing offspring. And this has improved the business. As an example, polo has a very complex breeding system. To produce animals, elite animals, differential animals, maybe you need to produce hundreds or 200 to have one animal with the traits that you're looking for. So this makes polo breeding very expensive. But with clones, with different copies of animals, of the best animals, when we breed those animals, the possibility that they will produce offspring with better genetic traits is much, much higher. This means that you need a smaller group of mares to obtain differentiated products. So the costs are reduced enormously because you need less animals, because all of the breeders are clones. Another characteristic that's very interesting is to recover the genetics of the neutered or castrated horses. 90% of the horses used in polo are the mares. There are some stallions that have good characteristics to be players too, but they must be docile. So they're castrated, the males, when they're young. When they arrive to their sports capability at the highest peak, you cannot create any offspring from those animals. So the genetics is lost because of the castration. But through cloning, you can recover those traits from the castrated animals. And a final advantage, a very important final advantage, is that you can come back to life animals from the past. Here are two clones that we produced in the lab at Kheiron from an animal that used to play in the United States in the 1990s. It was the best polo horse of the 1990s. It died, and the DNA was conserved. And a short time ago, some years ago, we were able to produce these animals again, bring them back to life. And what does this allow? What's the benefit of bringing animals that are no longer here, that we can breed them with current animals? These mares that were amazing horses 20 years ago can now be bred with stallions from today. And thus we have offspring that naturally would have not been able to create because they lived in different eras. So this is another great advantage, cloning the past with the present. And the first clone of a polo horse in Latin America was in 2013. And as of that moment, a real revolution started in polo. the era of clones, age of clones. Not only because clones were being produced, but Adolfo Cambiaso, which is the best polo player in the world, he began to use these clones in the year 2013. So this revolution of cloning expanded. And years went by, and Mr. Cambiaso, the player, started to use more and more clones on his team. Here's a very emblematic image of Mr. Cambiaso going from the original horse, there's Cuartetera, to the clone. This is 2014. He had more clones, he kept adding clones. And this is the list of horses from last year, from the Polo Open of Argentina in Palermo, a neighborhood, which is the most important tournament, polo tournament in the world with no limit in handicap. The best players here and the best horses play here. And you see on the same team, 7 clones of the same horse of the same animal. Year by year, clones have more acceptance. And I could also say, it's not only this team, which is La Dolfina, but today, the best 4 teams in the world are producing clones. And many other polo players are making clones. So we're going to start seeing in 3, 4 to 5 years, more and more clones. And you're going to see them more and more in the playing field, in the pitch. Why all this? Aside from the advantages that I mentioned. This produces a lot of advantage for the breeders, but it also has sports advantages. The animals, for example, the most important tournament played in Argentina, in Buenos Aires city. Once this tournament is over, then you choose the most valuable horse, like the most valuable player, the most valuable horse. In the last 3 years, the winner of this most valuable horse after the match has been a clone. So today, clones are definitely the best horses competing. But not everything is polo. We have also seen requirements or interest in cloning, such as the Equestrian European Federation and endurance race in the Middle East. Other sports, other equestrian sports, horses are starting to see the advantages of having a system of having their horses through cloning. But let's see. Now we're going to see a little the technical side, the scientific side. How is a clone made? Have the concept, the basic concept of how a clone is made. To make a clone, we need 2 raw materials, 2 main raw materials. One raw material is the DNA of the animal that you want to clone. To make a clone, you need a genetic reserve. And that genetic reserve is in the cells of the animal that you want to clone. The first thing you do is to make a gene banking. This reserve of DNA from the donor animal is called gene banking. How do you do this? The most common manner to make a gene banking is to take a small piece of skin of the animal that you want to clone with a special instrument. It's called a punch. It has a tip, 5mm tip in diameter. This is inserted into the animal's skin. For the animal is innocuous. It just leaves a little circle, 5mm diameter. And this tiny sample is enough. The amount of cells that are in this sample is millions. This tiny bit of skin is taken to our lab. This is This is a Petri dish plaque in which you fragment this piece of skin, you process, you put into a special incubator that emulates the physiological aspects of a mare, and the cells start to multiply and multiply and multiply by millions. Here's a microphotography of these cells. And afterwards, the cells are fragmented into small tubes, and they are cryopreserved in liquid nitrogen at 200 degrees below zero. Once they are at that temperature, the gene banking can last for decades, decades, and decades, and can be used when you need to make a clone. You can make the gene banking today, you can unfreeze 10 years from now, and those cells are totally viable for producing a clone 10 years later. That on one side. Two raw materials we need. The first is the DNA of the donor animal. The second one, oocytes. Oocytes. We need oocytes from a donor mare. As if the DNA was the instruction manual to make an animal, the oocyte is the machinery that reads the instruction manual and produces the embryo, and then a complete individual. Now, the oocyte has a particularity that it has its own DNA. So what we need to do is remove that DNA through a very complex process called micromanipulation. We remove the DNA, the chromosomes. So we have an oocyte that is blank, that we can put in the new instruction manuals of the DNA from the donor animal. The cells from the gene banking that we took from the donor animal. These donor cells are fused with the oocyte, and there we have an embryo. This is a clone embryo that is then cultivated, incubated in the lab. 7 or 8 days is the incubation type. The embryo progresses in the lab, where it reaches a status that is compatible with an embryo transfer into a receptor mare. So the clone embryo is implanted into the surrogate mother. It has to have certain characteristics. It's the one that receives the embryo, which was formed with the donor material. This mare produces the production of the horse, and after 11 months, the horse that is born is a clone of the first individual, where the DNA sample is taken from the skin. This is how you make a clone. Now, this procedure, which actually might seem simple like this, is very inefficient. Very inefficient. Above all, in a stage which was when this oocyte, you add the cell, the adult cell of the donor cell. Why? Because this cell is reprogrammed to be a skin cell. We are taking the skin cell and we're saying, stop being a skin cell and form an individual, a complete organism. This that the oocyte has to do doesn't do it well. This is a reproduction, anti-natural reproduction. So this oocyte is not prepared to do that. So there are a lot of errors. Why do I say this? Because this brought a series of disadvantages in the cloning procedure that we will see them now. Brought lots of deaths and malformations. So something important our lab has is an R&D program, a constant R&D program, very focused on the welfare and the equine welfare and the welfare of the horses. As I said before, in 2015, all the reports of birth of horse clones informed that about 50% of them had real complications, deviations in their forelimbs or in the navels, and even death. So we started to do more R&D. And as I told you, and the problem was in the origin of those cells, that you were using a skin cell. Skin cell has a lot of information to express itself as skin. No, and we say to that cell, no, don't be skin, be a complete individual. So what we did, this is a paper that we published some years ago. Why don't we start using bone stem cells, which are cells that have a pluripotential to produce different tissues, such as mesenchymal stem cells. So we started to produce stem cells from these mesenchymal stem cells, rather than skin cells that we obtained through the bone marrow. As of this moment, when we change the origin of the cells, which is the donor cell, the DNA, we started to improve the viability of all the clones and their health, the health of those cloned horses. At least 100%. Almost 100% of our animals are born perfect, while before, reported in all the labs around the world, was less than 50%. So we continued with our program of investigation, R&D, trying to, once we have perfected this process, we started with other technologies, which is a technology that I named a while ago, genetic editing. This is through CRISPR-Cas technology. It's a technology by which last year, two of the researchers involved in the development of this technology got a Nobel Prize. CRISPR-Cas allows you to set changes in the genome of the individual with a very strong precision. You can include new strings of DNA, eliminate the expression of certain DNAs, and you can even rewrite the genomic code in a precise way. We started to apply in horses. Why? Because, as an example, we know genetic sequences that can predict if a horse will be better for running short distance or long distance endurance. And in that, based on certain sequences of the genome, in this case, myostatin protein, for example, is a regulator of the muscular mass. So we began studying this gene, and last year we published the generation of the first horse embryos edited genetically, CRISPR-Cas, and we continued this research, and we are modifying certain characteristics of the muscles of the horses. This is still in an embryonic stage, no births yet, but we're very close to transferring these embryos to a surrogate mother. This news was followed around the world in the media and the press. It actually had the visit of the ex-Secretary of Agriculture of the United States to see the process, to visit the laboratory. He wants to know more about this technology and about our technology of editing, of genetic editing. Until here, this is us. Thank you for your time. And thank you for the invitation. And thank you for having paid attention to this explanation on cloning. Thank you, Gabriel, for your time and for sharing your knowledge with us. No, thank you for the invitation. If anyone wants to ask a question, please do it by chat or directly through voice.
May I ask a question?
¿Te puedo,
course.
Adi, traducís vos? Okay.
Hi, my name is Liviu Gordea. I'm a journalist from Romania. And first of all, congratulations for your presentation and to share with us, to have this opportunity to share with us your knowledge. And I would like to ask you, what do you think about or how do you see the future of animal cloning in agriculture or for agriculture animals?
Dale. Well, I believe that cloning has a great future in combination with genetic editing, the one we were talking about right now, because genetic editing can give new characteristics, differential characteristics to these animals. For example, adaptability to climate. I'll give you an example. We are in a development of genetic editing and cloning with a company from the US, which is called Agrocombinetics. We have a grant from the Bill Gates Foundation, and what he's interested is producing cows with differentiated characteristics that can be raised in Africa, that are high capacity milk cows that would resist tropical conditions, adverse tropical conditions. We are studying how to make those cows to produce more milk, that they have much shorter hair, and that they will resist higher temperatures, and they will be born without their horns so that they do not hurt each other. All these characteristics. Or, as I said, we are producing animals that conduce less to produce more protein, conduce less food to produce more protein, or that they resist certain foods, certain illnesses. All these characteristics that we can edit in the cattle and we can produce in one type of cattle are done with cloning. So I believe that the future of cloning in cattle will be there. It's a advantage combined with gene editing than cloning itself as duplicating animals. Cloning plus genetic editing.
Thank you.
Not just duplicating animals. Any other questions? Does anyone else have a question, please? You can say your question by voice. Okay, Gabriel, thank you very much once again. We have a question. Okay. Adi, traducís vos.
Yo no escuché, no, traducilo vos, George.
No sale el sonido.
I can put the question in chat.
Okay. No se
Oh, he
escucha,
can't
George.
hear
¿Eh?
me.
Estoy en el chat.
No, él tiene solo español. Debe estar en español. No, deja a Adi. Pregúntale, la pregunta si él piensa que dado el bajo costo de comparando una vaca con un caballo deportivo, ok. Yes, I believe that the value of a great horse or a polo horse is different from a bull or a cow or an important cow. Nevertheless, the prices of cows or the important breeds in Argentina is increasing. And we also have the advantage that the technology of gene editing is very simple and it's very cheap. It's not expensive. So I believe that's why it's going to be accessible for many laboratories. So the cost of transgenic editing and cloning is not expensive. So you can do it in the lab at a very good cost. We have already had companies, clients, commercial clients that want to apply to cattle. And in fact, we are working with many in principle with a commercial agreement with cattle breeders that want to improve the genetic traits of their breeds. So I think yes, there will be use because the cost will not be too high. Thank you. Now we say goodbye to you, Gabriel. Thank you very much. We appreciate your time. And if anyone wants to reach out to you, is there a way for them to write to you? Sure, please. In my chat or Instagram or LinkedIn, here's my email in the chat. If anyone wishes to have any additional information or reach out to me for any help or research, there's my email is gvichera@gmail.com. Thank you very much, Adi, and everyone for the invitation, for having listened to this presentation. And there's my email. That's my personal email. If not, social media or the page, the website, Kheiron Biotech. You can find me there. Thank you very much. Have a good day. Thank you. Goodbye to all. Thank you. Thank you. Bye. Goodbye. Was the sound okay? You're welcome. Thank you, Hugh. Thank you very much.
We'll get the recording edited and we'll put it up on the website in English and Spanish.
Great. Thank you very much. Have a nice day.
Okay, thanks.
Bye-bye.
Bye-bye. I'd take you out of the box, George.
A good day, Hugh.
Sorry, I have to take you out of the box.
Take me out of the box. Yeah, that was good. Yes, please.
Yeah.
I don't want to stay in the box all day. Thank you.
Okay, thank you.
Bye. Take care. Bye-bye. See you next time.
Bye-bye. All right.